Welcome back, everybody. We're just about to start our final session of the course. So until now, most of the focus of the lectures has been on later drug development, on clinical trials, particularly phase 3 clinical trials. And the session that we're about to begin deals with early drug development and the aspects of drug development that are included in the investigator's brochure, which you've all heard about several times in lectures today. So I think in understanding the investigator's brochure, it's very important to have an understanding of some of the basic non-clinical work that's done and some of the early clinical pharmacology. And to address those, we've got three excellent speakers. Paresma Patel is going to be talking about chemistry and manufacturing controls. We're going to move on to Matthew Thompson, who's going to tell us about pharmacology and toxicology. And then finally, we're going to end with a talk from Shirley K on clinical pharmacology. So let me hand it over to Paresma to begin.
Good afternoon. My name is Paresma Patel, and today I'll be presenting on chemistry, manufacturing, and controls requirements during early clinical development, with a focus on small molecule CMC development. I'm currently a division director within CDER's Office of Pharmaceutical Quality in the Office of New Drug Products. Today, I'll start off with a brief description of pharmaceutical quality, define some key terms, and then go through the CMC requirements that are outlined in the Code of Federal Regulations and further described within guidance documents for INDs. I'll discuss some specific requirements for drug substances and drug products, and I'll discuss key CMC information related to impurities, give a brief description of specification tests and information needed on stability. Finally, I'll end with a brief description of the main issues related to CMC that are often considered safety concerns and may lead to clinical holds. I'll end with some key guidance documents and resources as references. While this talk is focused on early drug development, the vision of the Office of Pharmaceutical Quality gives you an idea of the end goal of that development. We believe that a quality product of any kind consistently meets the expectation of the user, and drugs are no different. Patients expect safe and effective medicine with every dose they take. Pharmaceutical quality is assuring every dose is safe and effective, free of contamination and defects, and it is what gives patients the confidence in their next dose of medicine. I would like to start by providing two key regulatory definitions for drug substance and drug product as defined in the Code of Federal Regulations. The drug substance definition is shown here, and to summarize, the drug substance is the active ingredient. It provides the pharmacological activity or direct effect for the diagnosis, cure, mitigation, treatment, or prevention of a disease, and it is the active component of the drug product that affects the human body. The drug product definition is also provided in the CFR, and it is the finished dosage form that contains the drug substance, and it may or may not contain other ingredients. The requirements for CMC information for an IND are described in the CFR and provided here. Now, I won't read this exact definition, but I will distill this information into the basics and then discuss how the CFR has been interpreted as outlined in FDA guidance documents. The main text of the CFR can be broken down into a few main requirements for CMC information for an IND submission. They include the composition, manufacturing process, manufacturer, and description of how the drug substance and drug product are controlled. Information to assure identity, quality, purity, and strength of the investigational drug. It's important to note that the regulations give flexibility for the amount of data that should be submitted depending on the phase of investigation, the dosage form, duration of use, and any other available information. The emphasis for a phase 1 submission is really the identification and control of the raw materials and new drug substance under investigation. As you read further into CFR 312, you see that the law stipulates the data necessary for both the drug substance and drug product. For the drug substance, it requires a description of the physical, chemical, and biological characteristics. Again, this is very broad to allow FDA discretion to determine what is necessary. The sponsor has to state who makes the drug, how they make it, and provide sufficient information to demonstrate identity, strength, quality, purity, and stability. Similarly, for the drug product, the same general advice is outlined. The requirement is to provide the components of the drug product, the quantitative composition, information on who makes it, how they make it, how they package it, and information to demonstrate its identity, strength, quality, and purity, and finally its stability. Overall, I think it's clear that the CFR requirements are vague and pretty broad. So what I'll do next is go into a little bit more of the details of the information that CMC reviewers are looking for, specific to both drug substances and drug products. Now I will go through some of the drug substance or active pharmaceutical ingredient related CMC information that reviewers generally are looking for as part of the IND review process. I'll focus on the following topics as part of my presentation today: manufacturing process or the route of synthesis. Essentially, this is how the drug substance is made. Then I'll move on to the characterization data, which I'll cover in two parts: structural characterization data and characterization of the physicochemical properties. I'll discuss how the drug substance properties impact the drug product. And then I'll finally discuss the control and understanding of impurities, touch on release specifications, and finally the stability data that should be submitted for the drug substance. I'll start with the first two topics outlined here in red, and then discuss impurities, specification test, and stability in conjugation with the drug product CMC information. First, I'll start with the manufacturing process of the drug substance. Today, I'll focus on new molecular entities, or NMEs, because the manufacturing process for NMEs is often at an early stage of development when an IND is submitted. In order for us to assess the safety of the drug substance to be used in a clinical study, we ask for a brief description of the synthesis used to manufacture the clinical trial material. This information should be provided in both a written format as well as a scheme that provides all of the chemical reagents, solvents, and catalysts used as part of the process. As part of the manufacturing section, it's useful to provide any information on in-process controls. For example, are there any analytical tests that are run to ensure reaction completion at each step of a process? If the manufacturing process of any starting materials is available, this information is helpful for reviewers to assess potential impurities and provide further CMC developmental guidance. However, this information is not required for an initial IND submission. While not required, any available information for control of raw materials or isolated intermediates provides valuable information to reviewers to understand the points within a manufacturing process where there are opportunities to purge potential impurities. Because the synthesis is often being continuously modified and improved, there may be differences in the manufacturing of the toxicology and the clinical batches. If this is the case, it is important to provide the schemes for the toxicology batches and the clinical batches, and point out any specific differences in the routes and in the batch data. For example, are there any differences in process impurities, residual solvents, or elemental impurities? Many sponsors may choose to use the same batch for toxicology studies as they intend to use in the clinical studies, and this approach is reasonable from the FDA perspective. However, we remind you that the non-clinical studies should be supportive of the proposed clinical doses, and a sponsor may want to assure that there is adequate data to support any increases in impurities that are observed on storage. Next, I'm going to move on to talk about the characterization data that is expected for your drug substance. I'll group this into two categories: structural characterization data and physicochemical characterization data. First is the structural characterization data. This is the data that is needed to support the proposed structure of the drug substance. We recognize that structural characterization data for a drug substance may be limited early in development. As part of the characterization and to assure identity of the drug substance, we expect to see submission of multiple characterization techniques to support the structure of the drug substance. Some of the standard techniques for small molecules include spectroscopic data such as NMR, UV-Vis, or IR. We often see liquid chromatography techniques coupled with either UV or mass spectrometry as part of routine identity testing. As part of your characterization data, we expect submission of the raw spectral data. However, in addition to the raw data, we expect your interpretation of this data. For example, peak assignments for NMRs or functional group assignments for IRs. During IND review, we will then evaluate the totality of the characterization data and your interpretation independently and provide any comments or developmental phase-appropriate advice. We acknowledge that identification of some peaks may be ambiguous, as you may not have identified all of the impurities that are present at low levels. I want to briefly describe some of the physicochemical characterization that should be considered in the context of its criticality to the final drug product formulation and overall drug product stability. These types of attributes include physical attributes such as appearance and physical form of the API. Is the drug substance a solid or an oil, and what color is it? We want information on the solubility in aqueous solutions as well as organic solvents that may be used as part of the manufacturing process. Information on the particle size distribution is likely limited at an initial IND submission, but the potential impact of this attribute for the drug product should be considered as additional clinical batches are manufactured. A complete understanding of polymorphic form is not necessary in the original IND submission. However, it would be important for you to know the polymorphic form of your toxicology and clinical batches. And if different forms have been identified, forms should be monitored or controlled at release and on stability. It would also be helpful to know if polymorphic form is likely to impact dissolution, bioavailability, or stability of your final drug product. Hygroscopicity is the propensity for a drug substance to absorb water. Information regarding whether your drug substance is hygroscopic should be provided. We want to know if the drug substance is hygroscopic, what is the risk that this may impact the overall stability, polymorphic form, or bioavailability of the overall drug product. We generally encourage that sponsors monitor and characterize those attributes throughout development and assess the criticality of these attributes to the drug product, with the understanding that there may be changes to the drug product formulation in early development. I would like to now jump over to the drug product and clarify some of the specific information that is needed for a drug product compared to the drug substance for early clinical development. For the drug product, it is important that we have a clear description of the dosage form and route of administration. We would need information to justify the safety of any novel technology or complex drug product formulation. It's important to clarify any administration information that may be important for that dosage form as well. For example, the burden of data becomes greater for a drug product that has to be administered parenterally, as you have to address sterility and endotoxin concerns and consider any stability or compatibility issues associated with any administration devices or solutions for diluting the drug product. The quantitative composition of the drug product should be provided. That includes a list of the inactive ingredients or excipients used in the formulation. We ask that you provide information on the compendial status or quality of those excipients. If novel excipients are used, we would need additional information for the manufacturing and controls to assure the quality of those excipients. Additional information may be needed to assure the safety of any animal-derived excipients as well. The drug product manufacturing process should be described with a brief written description and a flow diagram, including available information for any controls implemented during the manufacturing process. Additionally, information on the sterilization process should be provided in this section if applicable. I'm now going to discuss the information related to impurities, specification tests, and stability in the context of both the drug substance and the drug product together. In the next couple of slides, I'll touch on the information related to impurities that we expect as part of an IND submission. Impurities are defined as any component of the drug substance or drug product that is not the actual chemical entity. Drug substance impurities can mostly be categorized into the following: organic impurities, or process-related impurities are those that arise during the manufacturing process or on storage. The specific thresholds for reporting, identification, and non-clinical qualification of organic impurities are further outlined in ICH Q3A for drug substance and ICH Q3B for drug products. Mutagenic impurities and the toxicological thresholds based on the duration of treatment are defined in ICH M7. I do want to point out that the scope of this guideline may not be applicable for advanced cancer indications or in cases where the drug substance is itself mutagenic. Residual solvents are those solvents that may carry through from the manufacturing process to the drug substance, and the acceptable limits are defined in ICH Q3C. Elemental impurities may arise from the reagents or catalysts as part of the manufacturing process, or from the manufacturing equipment. The acceptable limits and tests are defined in USP chapters 232 and 233 and in ICH Q3D. And finally, microbiological contaminants and limits are further defined in USP chapters 61 and 85. At early stages of development, we want to know what your control strategies are for all of the potential impurities and classes of impurities that I described in the previous slide. We do not expect that all impurities are necessarily identified as part of an initial IND submission, but we do want to know your proposed strategy to further identify and control impurities. At the stage of an initial IND submission, we ask that any differences in the impurities between toxicology and clinical batches should be addressed and justified based on the available information. For example, if there are impurities present in your clinical batches at greater levels than in the toxicology batches, is there sufficient non-clinical qualification data to justify the levels in the clinical drug substance? Or are the levels below the qualification threshold outlined in ICH Q3A? As part of the understanding of impurities, a list of identified process impurities or potentially mutagenic impurities should be provided. Additionally, a comparative list of the impurities by relative retention time between toxicology and clinical batches may be provided if identification efforts are ongoing. If there are non-clinical data to justify impurities present in your clinical batch, provide reference to that data as part of your justification. Additionally, if there is a potential risk for formation of mutagenic impurities based on the manufacturing process, provide information on whether those impurities have been identified. Additionally, you may want to provide some spike fate or PERS data to justify why the risk of impurities to impact your drug substance may be low. Now I'll move on to the overall controls for release of the drug substance and drug product. The specification is the set of criteria used to test and release batches of drug substance and drug product for clinical use. The specification table provides a list of the test methods and proposed limits to assure that clinical batches of drug substance and drug product meet the requirements assuring identity, strength, quality, and purity. The drug substance specification will have different tests compared to the drug product specification, but the overall format of the specification table is similar. In the initial IND, the analytical test methods can be briefly described or referenced to a compendial test. The limits for an IND are typically flexible based on available toxicology and clinical batch data, and are often further defined and narrowed as development proceeds and nears commercialization. As part of the IND submission, we expect to see either tabular batch data or certificates of analysis for the proposed clinical batches tested according to the proposed specification. We want to upfront acknowledge that the specification test and limits are likely to change through development. Often, the analytical methods are being developed and validated, and the manufacturing process is being optimized for efficiency, but also to remove toxic or dangerous reagents or solvents, and the final commercial process and scale is being developed. However, as development moves forward and additional batches are manufactured, we generally want to see that the specification, test, and acceptance criteria are becoming more well-defined and justified as development progresses. Finally, I would like to briefly discuss the amount of data that is acceptable for an initial IND when it relates to stability. Preliminary data on representative material for both drug substance and drug product is acceptable for an initial IND submission. If stability information on the clinical batches is available, that information should be provided. The stability section of an IND should clarify the tests that are run to monitor stability. If stability data is not available, we ask that sponsors provide a commitment and a protocol to continue testing the clinical trial material through the duration of the proposed clinical study. I would like to now touch on the major CMC safety issues that could lead to clinical holds. The likely concerns include any question related to adulteration or purity of the materials used as part of the drug substance or drug product manufacturing process. If the impurity profiles are not sufficiently characterized, or the safety of the impurities is not justified by supportive non-clinical studies. Any impurities that are known to be highly toxic or pose potential risks to patients. Unreliable analytical methods that may undermine the confidence in the data provided in the IND. Insufficient batch data to demonstrate manufacturability of either the drug substance or the drug product. Any significant stability issues under the proposed storage conditions. And lack of sterility assurance or endotoxin controls, for example, for parenteral drug products. So while the CFR sets legal requirements for drug applications, there is broad discretion for FDA to apply these regulations. There are resources to fill the gaps, including FDA and CDER guidance documents on a wide range of topics. These are openly available, and I've listed some of them at the end of the slide deck. There are also applicable regulatory standards such as the U.S. Pharmacopoeia and National Formulary Monographs. When standards aren't stipulated in the USP or NF, it may be appropriate to reference monographs from other regulatory agencies based on your scientific justification. And finally, I would like to point out that pre-IND meetings are recommended and valuable resource to get direct FDA feedback on your specific development program. And with that, I'd like to end and take any questions that you may have. I've provided here a list of other guidance documents and references that may be valuable to you.
Thank you for the introduction. So we just heard about product quality information to support early drug development. And now we're going to switch gears and discuss pharmacology and toxicology information to support early drug development. My name is Matthew Thompson, and I'm a supervisory pharmacologist in the Division of Hematology, Oncology, Toxicology at FDA. The objectives of my talk are listed here. Given the short time I have today to discuss pharmacology and toxicology information, it is important for you all to know where to go to find additional information, and that is in large part going to be through FDA guidances. Now, the bulk of my talk is going to be about the types of non-clinical information you can expect to find in the investigator's brochure for a product. And I will continue to come back to this outline here of non-clinical information during the talk. So one of the most important links I can provide you today is at the top of this slide. And at this URL, you're going to be able to search for FDA guidances using keywords. And then below is a short list of guidance document topics with a letter designating the type of guidance and the number indicating the sequence. So, for example, here S refers to safety, and M, or in this case M3, refers to a multidisciplinary guidance. So you may find guidances helpful to both understanding what information you're seeing in an investigator's brochure, and also, if you're involved in development, what kinds of non-clinical studies are expected to support a clinical investigation. So without getting too detailed, I'll briefly say that in addition to the S for safety and M for multidisciplinary, there are also guidances which are Q for quality and E for efficacy. It's also worth pointing out that there's a chance you're going to be investigating a product in a healthy volunteer population. And so the M3 guidance is particularly relevant if you're going to be developing this type of product. Whereas the S9 guidance is very relevant to products used to treat patients with advanced cancer. And lastly, given the number of biologics we're seeing that coming into the agency, the S6 guidance is also worth a read. Now I'm going to shift gears and discuss the non-clinical information one could expect in the investigator's brochure. So Title 21 of the Code of Federal Regulations, Part 312, deals with investigational new drug applications. And 312.55 states the sponsor shall give each participating clinical investigator an investigator brochure. So what is in an investigator's brochure related to pharmacology and toxicology? Well, 312.23 discusses the IND content and format, and in A52 it states that there will be a summary of the pharmacological and toxicological effects of the drug in animals and, if known, in humans. Well, that's not providing a lot of detail, so we'll go into that in a little more depth during this talk. But before we start, one thing may be worth noting, and that's related to the terminology that I will be using. So at FDA, the word non-clinical is often used interchangeably with pharmacology and toxicology. So here is the outline of the non-clinical information in the IB that I'll be covering today. Pharmacology, safety pharmacology, and toxicology topics including general toxicology, genetic toxicology, and other. First, we'll discuss pharmacology, and by this we generally mean information on the mechanism of action of a product and its activity, whether that be in vitro or in vivo. And we typically break that up into thinking about both primary, or the intended target and effects, or secondary, the unintended target and effects. So if we have a small molecule drug targeting a cell surface receptor, for example, I may anticipate certain toxicities based on this mechanism of action and how interfering with the receptor results in changes in signaling, leading to both the drug's therapeutic activity but also potential side effects. Primary pharmacology information may include binding assays, inhibition assays, data from cell lines, as well as the product's activity in an in vivo model of a disease. Secondary pharmacology will often consist of panels or screens involving many different enzymes, receptors, or ion channels to assess off-target effects of a product. And the type and amount of data varies based on a number of factors, including the product type. Is it a small molecule or a biologic? The indication, in other words, what's the patient population? Are we talking about a healthy volunteer study? Or are we talking about a population with advanced cancer where life expectancy is shorter and available therapies are limited? And finally, the stage of development could influence the type of data available in the IB based on whether the pharmacology data is important to determining a first-in-human dose versus maybe later in development where understanding a toxicity that's been observed in the clinic may be informed by certain pharmacology data. The next area of non-clinical information is safety pharmacology. Safety pharmacology refers to studies designed to assess the potential of a product to affect physiological functions. And in practice, we are most concerned with assessing the effects of a product on vital functions. So therefore, the core safety pharmacology battery that you can expect to see information on is cardiovascular, central nervous system, and respiratory, since these are vital functions where acute effects could be life-threatening. Cardiovascular assessments may include in vitro assays to study the effect of a small molecule drug on a cardiac ion channel, and ECG data from a non-rodent species. Central nervous system and respiratory safety pharm studies are often conducted in rodents and will involve assessments of behavior, other neurologic parameters, and respiratory parameters such as respiratory rate and tidal volume. Moving back to our outline, we're going to be talking about toxicology and the first topic, general toxicology. General toxicology studies provide critical non-clinical information, allowing us to determine whether a clinical investigation is reasonably safe, whether the initial dose proposed in a clinical study, in other words, the first-in-human dose, is acceptable. General toxicology studies provide information on the potential dose-limiting toxicities that may be observed in human subjects, and provide information on what to monitor for in the clinic. For example, a product may exhibit hepatotoxicity, so we may want to closely monitor liver enzymes and bilirubin, or if it was cardiotoxic, troponins, or if we anticipated effects on the immune system based on the mechanism of action, cytokine levels. And very importantly, general toxicology studies in animals allow assessment of potential toxicities that we cannot readily identify in a clinical trial, namely pathology findings. So before I give you an example, I want to briefly describe the components of a general toxicology study. Again, this is brief. So a general toxicology study will provide information on the test article, the test species, the dose levels, the schedule of administration of the test article, the route of administration, the formulation, and other aspects of the design, such as the species, the age, the sex, etc. And the outcomes of the study will include mortality, clinical observations, body weights, food consumption, ophthalmology, ECG findings, clinical pathology, which includes hematology, clinical chemistry, and urinalysis. There's also gross pathology, organ weight data, and histopathology. When FDA reviews all of this information, we are looking at the totality of the non-clinical data, both the pharmacology and the toxicology, and we are assessing whether or not a proposed starting dose is acceptable and what safety signals need to be monitored for in the clinical study. So here's our first example. This is a biologic. It's an antibody drug conjugate being developed for the treatment of cancer. And in the animals, there was significant myelosuppression and severe hepatotoxicity that included both increases in liver enzymes and histopathology with finding of necrosis. So in a clinical trial, the labs would provide the information on the liver enzymes, but may not clue the investigator in as to the severity until it's too late, and the dose of the drug maybe has been escalated too high to a point where there's severe toxicity. Because we have the histopathology component from the animals, we can understand if the product causes a pattern of toxicity in a particular organ that includes that pathology, such as the necrosis, and tells us to be particularly careful in considering how the protocol is written. For example, knowing the potential for severe hepatotoxicity may affect the inclusion and or exclusion criteria for the patient population in the trial. In considering the relevance of the non-clinical study to predicting toxicity in a clinical study, we consider two important things. The first is the design of the general toxicology study, with consideration of the route of administration and the schedule. And we're looking for that to be consistent with the clinical study. So for example, this is intravenous versus oral administration versus intramuscular, etc. And that this should match the planned clinical study, and the schedule should be consistent as well. So every day dosing, every other day, every week, etc. So it's important that the general toxicology study design provides the most valuable information. And consistent with this idea is the relevance of the non-clinical species. Typical non-clinical species used for evaluating products in general toxicology studies include two basic categories, rodents and non-rodents. So rat is the most common we see in the rodent group, followed by mouse. And typical non-rodent species are dog and monkey. So one thing sponsors will consider in the development of their non-clinical program and in the design of the general toxicology studies is what non-clinical species are relevant. And by relevant, I mean pharmacologically relevant. So a major consideration may be whether the target is present in that particular non-clinical species. And is the product going to interact with that target if it is present? We often run into this issue in dealing with biologics because they're so specific for targets, and a lot of the targets are not always found in rodents. So it's important that the selection of a non-clinical species is well justified and based on the science of both the target and the product. If the non-clinical species is pharmacologically relevant, then we are more likely to gain valuable information that can inform the clinical trial design, the investigator, and the patient about the risks associated with an investigational product. In this example, the small molecule drug, a microtubule inhibitor, is being developed to treat patients with advanced cancer. And this small molecule drug produced irreversible optic nerve degeneration in a rat study. As a result, clinical monitoring was increased, and information on this toxicity that was observed in rats was added to the IB and to the informed consent. And in this last example, a small molecule epigenetic targeting drug was found to produce malignancies in a 3-month repeat dose toxicology study in rats. The signal was also observed in the clinic, and appropriate steps were taken to address the finding. In this case, considering the patient population, 2 of the steps taken were the addition of the information to the IB to alert investigators and addition of the information to the informed consent to alert patients. The next topic in our outline is genetic toxicology. The area of genetic toxicology refers to the assessment of a product's potential to cause genetic damage. Since results from carcinogenicity studies can take a considerable amount of time to become available, genetic toxicology studies can be seen as a surrogate for carcinogenicity while clinical trials are ongoing, and this helps address concerns about risk to human subjects. A typical genotoxicity battery includes a test for gene mutation in bacteria, an in vitro chromosomal damage assessment in mammalian cells, and an in vivo genotoxicity evaluation. Timelines for genetic toxicology information to become available can vary. Typically, a gene mutation assay will support the determination of whether a single dose clinical study is acceptable. An additional in vitro chromosomal damage assay if proposing multiple doses in a clinical study, and a complete battery prior to phase 2. However, this can differ based on the indication and patient population. For example, if products are being developed in the oncology space, the full genotoxicity battery may be submitted with the marketing application. Now we're on the last topic, and though there are various toxicology studies that could fall under this other category, I'm going to briefly mention reproductive toxicology. An unfortunate example of drug-induced reproductive toxicity is thalidomide, which underscored the importance of using non-clinical models to assess potential for reproductive toxicity in humans. In considering the type and amount of non-clinical reproductive toxicology information in a regulatory submission, you can consider the patient population as well as the type of product. So is it a small molecule or a biologic? And you could even consider things such as the target or the mechanism of action. Reproductive toxicology studies covering fertility, embryo-fetal development, pre- and postnatal periods, you can generally look to the M3R2, S5R3, and S6R1 guidances. But based on the patient population, for oncology indications, you can also consult S9 and other FDA guidance. So to wrap up the talk, as you can see, we've really only kind of skimmed the surface of the pharmacology and toxicology information in an investigator's brochure. And in the non-clinical package that a sponsor will submit to the agency, there will be a lot more information. So if you'd like additional information on the types of non-clinical data that can be expected in a regulatory submission, you can consult the guidances and use that search function at that FDA website that I referenced at the beginning of the talk. Now, the pharmacology and toxicology, or the non-clinical data generated for an investigational product, is critical to taking an idea from the bench to the clinic, and from the clinic to an improved product that has activity and is safe. The mechanism of action of a product and its activity against a target considering off-target effects, effects on vital functions, and the product's overall safety profile as assessed by general toxicology studies, as well as other types of toxicology studies, lays the groundwork for the product's development program, moving it from the bench to the clinic. Here's the first challenge question, and it's based on a guidance I've referred to several times during the talk. What FDA guidance is most relevant for non-clinical drug development in oncology? A, M3R2, B, S1B, C, M7, or D, S9? The answer is S9. And the second challenge question. The safety pharmacology core battery includes A, cardiovascular system, B, central nervous system, C, respiratory system, or D, all the above. The correct answer is D. All three of these systems form the core of the safety pharmacology battery. Thank you.
Thank you, Leonard, for the introduction. Hello, everyone. Today, I'm excited to share with you all the cutting-edge scientific discipline of clinical pharmacology. Now, you may be wondering why clinical pharmacology is important, or maybe even exactly what it is. Well, what you will learn today will help give you a basic understanding about various aspects of this discipline and how it plays a critical role in almost every clinical development program. So let's get started. Here's my disclaimer. First, we have a few objectives today. We are trying to understand what clinical pharmacology is and also understand about its role in early drug development. So how will we get there? Well, first, I'll define what clinical pharmacology is and then give a broad landscape of what these early clinical studies look like. A lot of important details will be contained in here, including the timing of these trials, the goals for each type of trial, key design elements and information gained from these studies or trials, and also at the very end of the presentation, I'll give a little bit of information about model-informed drug development. So first, what is clinical pharmacology? Well, in short, it is the study of pharmacokinetics, or PK, and pharmacodynamics, or PD, of a drug in humans. And PK is simply just what the body does to the drug. And you may have heard the concept of ADME, which is absorption, distribution, metabolism, and excretion. And that's shown in the graph on the right, where the concentration of the drug is plotted against time in a concentration time profile, essentially visualizing what the body is doing to the drug over time. Pharmacodynamics, or PD, is exactly the opposite. We're trying to visualize what the drug does to the body, and that's represented by an effect over time profile, as you'll see in that bottom graph. And when you combine those two concepts together, we get a relationship called PK-PD, or sometimes exposure-response relationship. And that is when we combine a concentration time profile with an effect over time, we're getting an effect over concentration. So essentially, we are trying to quantify the amount of effect that's produced by a particular concentration of drug. Now, you may be wondering, what is the role of clinical pharmacologists throughout the drug development process? Well, essentially, we own the dose. We determine how to give the drug, how much of it to give, how often to give it. Every aspect surrounding the dosing regimen of a drug is something that a clinical pharmacologist helps to determine. We like to say, are we giving the right drug at the right dose, at the right time, to the right patient? And this is a very good simplistic summary of exactly what clinical pharmacology does. First, I'd like to go over the properties of a drug And most of you have heard of ADME. And the first letter, of course, stands for absorption. And here we're really trying to measure the bioavailability of a drug. And that is, once it's been administered to the body, how much of the drug are we able to quantify in the various biological matrices? It's usually blood, but it's not always the case. And also we're trying to look at the PK variability. Does it exhibit linear PK? Does the drug accumulate over time? Is the drug exposure significantly affected by various extrinsic factors such as food or medications, concomitant administration of grapefruit juice has been known to affect certain drugs' PK characteristics, and alcohol? Can it be affected by transporters? These are all important questions that we aim to answer as clinical pharmacologists. Next, we have distribution. We aim to find out whether the drug is reaching its target site of action, and does it reach it at effective and non-toxic concentrations? Does it accumulate in non-toxic or non-target organs as well? How much does it bind to plasma proteins? What is the extent of protein binding? And that is a very important concept, since only the unbound drug is considered pharmacologically active. Sometimes we recommend measuring unbound drug when we need to interpret certain types of data. It can also be important to know if certain drugs are reaching the brain or the central nervous system via the CSF. Next, we have metabolism and excretion, and these are often combined together in termed elimination. We are trying to find out if a drug is metabolized to a great extent by cytochrome P450 enzymes or through other enzymes. Is the clearance highly variable? Does it depend on other covariates such as age, race, gender, or comorbidities? This is important for us to know so that we know if intrinsic factors can impact how a drug behaves in the body. Is the clearance time dependent? All of these are very important questions that clinical pharmacologists aim to try to answer. There are other considerations as well. Is the drug a narrow therapeutic index drug? If yes, that means that very small changes in the drug concentrations could very significantly impact the efficacy or safety of the drug. This sometimes may require therapeutic drug monitoring in clinical trials or in clinical practice in order to help minimize toxicities or lack of efficacy. It's also important for us to know whether this drug could impact other drugs when given concomitantly together. As you know, many patients take more than one drug together. Could this drug be impacting the enzymes of other drugs that are metabolizing other drugs? In this case, a drug interaction evaluation may be needed. Next, I will move on to early clinical studies. And a quote I recently read was that if you can't measure it, it doesn't exist. And I think this applies to clinical pharmacology because the crux of everything we do in clinical pharmacology relies on our measurement of drug in the body. First, a little bit about the timing. Often we see clinical pharmacology studies being conducted throughout all different phases of drug development. That is not typically ideal. What's ideal is to front load some of these studies so that we get a very good picture of how this drug behaves, what are the PK characteristics, and it can help contribute to dose finding for phase 2 studies, dose finding for phase 3 studies, of course. A lot of questions that need to be answered in order to make a go or no-go decision in clinical development relies on clinical pharmacology information. So consider that in drug development, that a lot of these clin pharm studies should be done very early. At the very earliest, speaking of early, is the first in human studies. So what that is it's determined by preclinical studies usually and some tox studies. It usually looks like a randomized, placebo-controlled, healthy volunteer study. In some cases, they can go straight into patients, and those cases are fairly rare. What are we looking to gain from this first-in-human study? Well, we want to take a preliminary look at the safety and tolerability of this particular drug. Want to find out if there is a maximum tolerated dose. We want to see general PK characteristics such as variability, linearity, some steady-state parameters, and also some preliminary exploration of how this drug is eliminated. Next, we have an ADME study, and I've gone over what ADME stands for already, but the objective is to really understand the full clearance mechanisms of the drug and its metabolites, if applicable, in humans. And hot off the press, we just had a new FDA guidance on mass balance studies that came out this year. So a lot of times an ADME study is called a mass balance study. You start off with a radio-labeled drug molecule. We generally use a single dose in healthy volunteers at the intended route of administration. And then in the end, we're measuring concentrations of the parent and metabolites, all metabolites, to determine the amount of radioactivity in plasma, urine, feces, basically collecting all biological matrices to try to determine the total drug excreted. What we're looking to learn is overall pathways of metabolism and excretion of a particular drug, identify any and all circulating metabolites, especially ones that might be major or pharmacologically active, and determine the abundance of those metabolites relative to the parent or total drug-related exposure. And this could come in very important later on in drug development. Next up, we have bioavailability studies, and we also have a new guidance for industry on these types of studies posted already. The objective here is really just to learn the basics about a drug. You know, when is its peak concentration? What is its peak concentration? What's the extent of absorption of the drug? That's measured as AUC, or area under the curve. And sometimes it's compared to a reference formulation. Typically, these studies are crossover designs, single dose in healthy subjects. We're trying to measure the extent and rate of the parent drug and any major active metabolites, if that's applicable. You can assess a relative bioavailability or absolute bioavailability. Relative would just be one formulation against another. An absolute bioavailability study is only against an IV formulation. So an IV formulation is assumed to be 100% bioavailable since it goes directly into your veins, directly into systemic circulation. We can call that 100%. You can do a relative bioavailability study that tests, let's say, an oral formulation against the IV formulation, and that'll give you the percent of total absolute bioavailability of that oral formulation. What we're trying to gather here is a comparison of the amount of drug that reaches systemic circulation from each tested formulation. So very simple here. Next up, we have a food effects study. So this is also, we also have a very new guidance on these studies as well. The main objective here is just to find out what is the effect of food on the rate and extent of drug absorption from a particular formulation. I think we've all had experiences with taking medications with food, or sometimes it's told to us to take it without food, maybe at bedtime, in the absence of taking any food. Why is that, and where this information come from? You know, it comes from studies just like this, where we're taking a single dose in healthy subjects using the highest therapeutic dose of the drug. The fed state should be a very specific high-fat, high-calorie meal that we put into our guidance as an ideal, I guess, sort of high-fat meal. Other meals can also be studied, but they should try to match the caloric and fat content as closely as possible to our guidance. The PK assessments that come out of this study are very similar to a bioavailability study. And you can claim there's no effective food if the 90% confidence interval of the fed versus the fasted Cmax and AUC ratios are contained within 80 to 125%. And that comes from the standard bioequivalence criteria. And also, if it falls outside of this range, the clinical significance of the food effect would be determined based on the drug's exposure response profile. Recall that in a previous slide I said that this type of PK/PD or exposure response relationship would come in handy later, and this is one of the reasons why. So some very important information that we would gather from a trial like this is how to administer drug in future clinical trials, or we may be at the point of labeling instructions on how to administer the drug in patients with respect to food, which is also a very important concept. Next, I'll move on to organ impairment studies, and these have been written about in guidances online as well. So I won't go into a lot of detail about them, but what I will talk about is when these studies are important to conduct. So first, for a hepatic impairment study, we're looking for drugs that are given in a chronic condition and also systemically available. We also are looking to see if we know that hepatic metabolism or excretion accounts for a substantial portion, which is greater than 20% of the absorbed drug, of the elimination of apparent or active metabolite. So does the liver metabolize more than 20% of the drug? And if it's a narrow therapeutic index, it's irrespective of the proportion that's metabolized. We will probably recommend that you do a dedicated hepatic impairment study. Finally, if the metabolism route is unknown, we will recommend a hepatic impairment study as well. Next up is the renal impairment study. And when should one of these be performed? Well, very obviously, when the drug is likely to be used in renally impaired patients, and when impaired renal function is likely to alter the PK of the drug or active metabolites. Finally, and this is somewhat of a newer addition to our guidance, is any therapeutic proteins or peptides with a molecular weight less than 69 kilodalton. And this is based on historical data that we have. Another very important type of study is drug interaction studies. And not every drug needs to have multitudes of drug interaction studies. We start by sort of a stepwise approach to try to find out if a drug interaction study in humans is necessary. And it starts with in vitro studies to determine if the drug is a substrate for or maybe a modifier of very common drug metabolizing enzymes. Once we find that out, we might need to we might recommend you conduct an in vivo drug interaction study just to confirm those in vitro test results. We don't always suggest that, but sometimes we do, especially in cases where we see a positive signal in the in vitro test or the signal is very borderline. If it's clearly negative, we would not ask for one of these in vivo studies. Once we get the results of that those in vivo studies, those go directly into the labeling for the drug that would help to inform healthcare providers and patients on how to take the drug. And based on the results of those studies, you may end up with something that's like a dose reduction of a drug, or all the way up to a contraindication, which basically says in no circumstance can you ever give these two drugs together. Some key points to consider for drug interaction studies are that you have to really take into consideration, are you maximizing the possibility of detecting an interaction? So you want to make sure to very carefully look at the dose of the interacting drug, what is the route of administration of it, the timing of the co-administration, and how long you're going to be giving those two drugs together in the trial. And the degree of the effect is typically classified by a change in the substrate AUC. Again, the exposure-response relationship is really playing an important role here to assess what is the clinical relevance of this change in AUC. Sometimes we see a change of 25%, but based on what we know about the exposure-response relationship of a particular drug, we may declare that this is not an important interaction and we just allow these two drugs to be given together as per normal dose. That's one option. And for some drugs, a 25% change in its exposure is not tolerated based on either safety events or lack of efficacy. So again, we're looking at the exposure-response relationship very carefully. Another concept in clinical pharmacology that has risen to prominence in the last decade or so is physiologically based pharmacokinetics, or PBPK. Some of you may have heard of it. What exactly is it? Well, on the right-hand side shows a very nice simplified diagram of the key components of PBPK, and that is we are trying to see what intrinsic and extrinsic factors are doing to the various model components contained within PBPK. And it's very biologically or physiologically based, as you can tell based on the diagram and the name. We're looking at lungs, blood, kidney, liver, intestines, all these bodily organs and how they contribute to this model. So it is a mechanistic modeling approach. We basically input preclinical and in vitro, sometimes even in vivo data to help us predict the behavior of drugs. In the end, what we're trying to do is get a prediction of what the effect of a certain intrinsic and extrinsic factors would be on a particular drug's PK. A program we started recently in the Office of Clinical Pharmacology is model-informed drug development, and that really helps with early dose selection. If we have a well-timed and well-designed dose-finding studies, this will help avoid dose selection pitfalls and failures later on in development. We initiated this MIDD program to allow sponsors to meet with the review team that's led by clinical pharmacology. We grant about 1 to 2 meeting requests per quarter, and we generally prioritize selecting requests that focus on dose selection or estimation, clinical trial simulation, or predictive or mechanistic safety evaluations. So these are all sort of our top selection criteria in order to meet with sponsors. And if you need any additional information about this, you can just Google MIDD and FDA, and it'll take you straight to some information on our website. And now for some challenge questions. The first one is, true or false? Pharmacokinetics is the study of what a drug does to the body. The answer is false. Pharmacokinetics is the study of what the body does to a drug. The next question is, which is not an example of an intrinsic patient factor that could affect the pharmacokinetics of a drug? Is it weight, smoking, age, or genetics? The answer is smoking. Weight, age, and genetics are all intrinsic factors because they are intrinsic to the individual, whereas smoking is something that comes from external to the patient and therefore is an extrinsic factor. The last question is, true or false? ADME stands for absorption, distribution, metabolism, and excretion. The answer is true. And that is all I have for you today. Thank you for your attention. And I hope you've learned a lot of the basics of clinical pharmacology and how it can really help you in early drug development. I'd like to acknowledge Dr. Kelly Reynolds in the Office of Clinical Pharmacology at FDA, and of course Dr. Leonard Sachs at FDA. He's really been instrumental in putting this course together. Thank you.
Well, thank you very much, Shirley. Thanks, Matt, and thanks, Paresma, for a real tour de force in early drug development. I think you've done a remarkable job of covering a very complicated and important area. So let's move to some of the questions. I think let's start with some of the first questions for Paresma. Let's see. The first question is about impurities in the API stability to be reported to FDA. The question is how and when impurities in API stability should be reported to the FDA. Do you want to talk a little bit about that, Paresma?
Thank you. Yes, I'm happy to cover this. I think there was a question kind of focused on reporting of out of specification results to the FDA and whether they should be reported as part of the stability studies. And the answer is yes. It's actually very important that all out of specification results be reported to the FDA for drug substance and drug product, both during release and if they've been placed on stability. And with that being said, I would just want to point out that FDA understands that sponsors are really at an early stage of development, and they're trying to understand the best conditions for maybe storing for storage of their drug substances and drug product. And so there may be out of specification results, maybe, for example, in your accelerated study, or even in your for some of your supportive study in your long-term conditions. And you may want to decide to then revise your stability conditions in order to accommodate some degradation you may be seeing, or some sort of change in quality attributes that you're seeing. So we do understand that, and it is it is part of your quality management system that these changes that you see on stability be reported to the FDA. And usually for stability reporting, we ask that these results be updated in your IND concurrent as your clinical study is being run and as additional data becomes available. Thank you.
Thanks very much. There are a couple of questions for Matthew. There's one about guidances. Where does one look for guidance for radioactive therapies? Maybe you just want to talk about that guidance and where individuals can find it. Wondering if Matthew's having difficulty connecting. All right, perhaps let's move on to a question for Shirley, and hopefully Matthew will be able to connect in a minute. So let's see, Shirley, there's a question about when should a mass balance study be run, phase 1, 2, or 3? You want to take a crack at that?
Yes, thank you, Leonard. To best inform drug development, we would definitely recommend the mass balance study be performed as soon as feasible. That generally means in phase 1. Most of our programs will see mass balance studies conducted during that time, and that's for good reasons. We want to be able to characterize the drug's ADME characteristics as soon as possible to help inform future studies.
Good. I'm just trying to figure out if Matthew is back. Well, I guess let's get back to Paresma here. A lot of questions for you. Let's see. According to FDA, what kind of drug product, GMP or non-GMP, has to be used for preclinical and clinical phase 1, 2, 3 trials? Did you get that question, Paresma?
Yes, thank you for this question, Leonard. This is a great question because I think there's a lot of confusion around the GMP requirements related to drug substances and drug products during investigational studies. So I'll start by pointing out that the regulations that govern current good manufacturing processes, or GMPs, are outlined in the CFR. They're outlined in 21 CFR 210 and 211. And these regulations are really focused on commercial manufacturing of drug products and drug substances, and may not be appropriate for phase 1 clinical studies. So the FDA actually published a guidance that's titled GMPs for phase 1 investigational drugs. And so that really focuses on the requirements in 3 general areas that we're looking for and considering when looking at phase 1 investigational study drugs. And that is kind of focused on well-defined written procedures, controlled equipment and manufacturing environments, and then accurately and consistently recorded data at those facilities. And it's really the sponsor's responsibility to make sure that these things are being documented and that phase 1 clinical investigational drugs comply with these recommendations and this guidance. Moving on to the preclinical studies, there's no requirement that the drug substance or drug product that's used in any of the toxicology or preclinical studies are manufactured under GMP, and oftentimes they may not be. We understand that from the FDA perspective that those may not be. However, as part of your IND, we do recommend that you provide a very clear comparison of the toxicology batch that was used and any differences in the manufacturing process for the toxicology batch versus the proposed clinical batch. And then finally, moving on to the phase 2 and phase 3 manufacturing, those it is clearly spelled out again in that phase 1 guidance that phase 2 and phase 3 manufacturing is subject to the parts of 210 and 211, so are subject to those CGMP regulations. I hope that answered the question. Thank you.
So that covered a lot of the early drug manufacturing concerns. There's another question here for you, which I think covers the later stages of the drug. In other words, how long should the stability testing cover? Any thoughts about that, Paresma?
Yeah, thanks, Leonard. So I think this is a really good question and kind of goes back to what's required for an initial IND is oftentimes it really just depends on kind of what is available. We typically like to see some sort of supportive stability data. It may not be the data for the clinical batches necessarily, but data, some initial stability time points for maybe even your toxicology batches or some representative material that you manufacture for both the drug substance and drug product. If that's not available, oftentimes we do understand and we would want to see your stability protocol. Now, as you move through development, what we expect is that that stability data is continuously being updated and your IND is updated with the stability data that you're collecting. And so you're kind of doing those studies for the clinical batches concurrently with the clinical studies. And I do want to point out that we don't really establish a retest period during an IND development program. We don't expect a retest period, and we just expect that you're actually monitoring all your clinical batches concurrently with the studies. So usually by the time you kind of, again, that really depends on the duration of use of your study drug. So the amount of stability data that's required really depends on that. But we typically, by the time an IND is open for a year or two, we would expect to see the 6 months of accelerated stability data that's recommended in the ICH guidances, and then a long-term stability data depending on what the data looks like and how long you're planning on using that drug substance.
Thanks very much. I'm happy to see that Matthew is back. So, Matthew, I have a question for you. The first one is, where does one look for guidance on radioactive therapies? Any comments on that?
Yeah, so if you're going to be developing an oncology therapeutic, there's a recent guidance that was became available in August of 2019. It's entitled Oncology Therapeutic Radiopharmaceuticals, Non-Clinical Studies, and Labeling Recommendations, and that's available on the FDA website.
Great. And while you're on, another question for you. What kind of non-clinical safety studies are recommended for complex oral dosage forms already approved active ingredients? A bit of a complex question. You can unpack it for us.
Sure. So let's assume this is for a small molecule drug. So in other words, it's a product that would be approved as an NDA. So that's a change in formulation, sounds like. So we would generally think of that as an application covered by Section 505(b)(2) of the Act. So this is where at least some of the information required for the approval comes from studies not conducted by the applicant and for which the applicant has not obtained a right of reference. So the actual extent of the non-clinical package would depend on several factors, such as the indication, the excipients used in the formulation, the new formulation of this approved API. So FDA would consider the proposed listed drug and the proposed non-clinical studies that would the sponsors or the applicants proposing to establish that a bridge between the information that they're going to reference and their new product.
Thanks very much, Matthew.
Sure.
Shirley, a couple of questions for you. Here's one on food effect. Is the food effect study done for all formulations, for example, immediate release or modified release, etc.? Any thoughts?
Thanks. Yeah, thanks, Leonard. In fact, it is recommended to be done for immediate release and modified release. Pretty much any oral product, we would recommend an evaluation of the effect of food on the absorption of that drug. Of course, this wouldn't apply to drugs that are administered through a non-oral route, such as IV or IM injections. But anything that goes through the oral route, we would recommend a food effect evaluation.
Yeah, that certainly sounds very sensible. Shirley, another question for you. When is it appropriate to conduct a reduced PK design for renal impairment? They give an example, only enrolling healthy versus severe population. Any thoughts about that?
Yes, and and I forgot to mention with the food effect, the guidance, the newest version, we just published it about 6 months ago, so that's available online. And for renal impairment, we also have a very recent guidance published just, I think, a year or two ago. And we do talk about the reduced study design. For those drugs that are eliminated via non-renal routes, a drug that we would expect is not, significantly renally excreted, sponsors can pursue a reduced study design, which is studying just the severe patients, starting off with those first.
And I guess while we're on the topic, another question about calculating the EGFR. Does the agency recommend a specific calculation for EGFR? And they give a couple of examples of calculations. Mm-hmm.
Right. To my knowledge, we don't make a specific recommendation. In the guidance itself, we have a table that describes the range of EGFR values for each category of function. But it is sort of, the onus is on the investigators, whether, they use Cockcroft-Gault, which is, of course, very commonly used in clinical practice. We have seen a rise in the use of MDRD calculations, but, we will accept either one. And sometimes we do an analysis on our end to kind of compare the two. Thank you.
Great, thanks. Back to Paresma. There's a long question here about particulates. Let's go through this. For appearance evaluation of an investigational drug product intended for intravenous administration, is there an expectation that visible particulates will be evaluated as part of the stability testing and reported within the IND during the initial filing? If not, at what point is this evaluation expected to be documented in the data provided?
Thank you for that question. Yes, in the short answer, yes, we consider particulate matter testing to be very important, particularly for parental administration, because it poses a potential patient safety risk. So generally, we would require that particulate matter is a release specification test, as well as it is tested on stability. And it is particularly important that it is very important that it be tested on stability, on your stability protocol as well, because we want to make sure that you're kind of looking for any aggregation or any precipitation, anything like that may be happening with your drug product. And there is a USP Chapter 788 that kind of outlines the particulate matter testing and the acceptance criteria around that we generally expect.
And there's a related question on impurities. Let me read you the question, and again, you can unpack it for us. In terms of impurity, there appears to be a consensus that ICH guidelines do not need to be strictly followed for early phase trials, especially for oncology investigational drugs. What about stability? Are sponsors expected to follow the ICH Q1E when setting retesting dates for early phase development? So the question is about impurities and stability.
Thanks for the question. So I'll start with the question about stability in ICH Q1E. Kind of going back to an earlier answer I gave, we don't expect a retest period is set at the time of an IND submission. My understanding is there are other regulatory authorities that do recommend a retest period, but the FDA does not. For an IND, we're really just asking that you submit the available stability data and any supportive stability data, as well as a stability protocol. Now, going back to the part about the requirements and whether ICH guidelines apply for early phase trials. It is true that the ICH guidelines that govern impurities, particularly ICH Q3A and Q3B and ICH M7, do not have to be strictly followed when setting limits for impurities during early development. However, we do recommend that sponsors consider the general principles of these guidance documents when considering impurity limits and the available information for identification and qualification of impurities. At the time of an IND submission, we're really, again, focused on the differences between the impurity profiles for toxicology versus clinical batches, and ask that sponsors really outline those differences between the toxicology and clinical batches and provide a justification for why any impurities that are in the clinical batch that are higher than the toxicology batch, how essentially justify the safety of those impurities based on what may be known about those impurities, whether any structural identification has been performed or whether those might have been qualified based on the non-clinical studies.
Great. Well, thank you for a very comprehensive answer there, Paresma. So I think it looks like we've run out of time. And again, I wanted to thank our speakers for a great session, a real tour de force through an enormous amount of territory, and thank the audience for excellent answers. So at this point, this brings our course to an end. It remains for me to express my enthusiastic thanks to all the people who've helped me put this together, both this year and over the years. In particular, I wanted to thank Brenda Stoddart and her team from the Small Business and Industry Assistance Program. They've done a tremendous job in coordinating all the different aspects, including the technical aspects, the CME aspects, and some of the content aspects as well. So thank you so much, Brenda. A fantastic job. I wanted to thank my colleague, Millie Dougal in my office, who's helped me tremendously with organizing the content of the course and doing the correspondence with many people involved. A huge vote of thanks to our wonderful speakers. I think the audience will agree that we really have a top-class group of speakers representing FDA's position on a whole variety of subjects. And finally, our most grateful thanks to a really excellent and enthusiastic audience. We saw a tremendous amount of audience participation. It's wonderful to see such interest, both locally and from all over the world. We really applaud you for your commitment to such a major public health issue, which is the development of new therapeutics. And we hope this course has been of some help to you as you move ahead with that career. And at this point, I just have to say that we're looking forward to our next year course. Hopefully that'll be in person and we'll be able to get to see each other face to face. So thanks again to everybody, and we will close the course at this point. Thank you.